So good day everyone, my name is Nicole Hanayu Aureliano, a member of the Group 1, and these are the members, Ms. Kaalaman Juvel V, Ms. Dagyo Cassandra Micaela, and Mr. Mabalio Ryan Ibrian. And today we are here to discuss and present about the biosafety in the laboratory. So now let's start the discussion. So first, what is the biosafety? Biosafety is the prevention of large-scale loss of biological integrity focusing both on ecology and human health. So in medicine, it refers of the levels of lab containment protocols measured as biosafety level 1, level 2, level 3, level 4 in order of danger. So the biosafety levels. Biosafety levels precautions to be taken by people researching trying to identify organisms. A biosafety level is the level of the biocontainment precautions required to undertake while handling dangerous biological agents in an enclosed facility. So what is containment? Containment safety methods, facilities, and equipment for managing infectious materials in the laboratory environment where they are being handled or maintained. The purpose of containment, reduce or eliminate exposure of laboratory workers, other persons, and the outside environment to potentially hazardous agents. The barriers. The barriers prevent invaders from crossing. Next is the universal precaution. Universal precautions is an approach to infection control to treat all human blood and certain human body fluids as if they were known to infectious of HIV, HBV, and other bloodborne pathogens. So what is personal protective equipment, also called the PPE? PPE is a protective clothing, helmets, goggles, or other garments or equipment designed to protect the wearer's body from injury or infection, specified by exposure, control plan, or by standard operating procedure. So what is Biosafety Cabinet, or so-called ABC? It also called as Biological Safety Cabinet or Microbiological Safety Cabinet. It is an enclosed, ventilated laboratory workspace for safely working with materials contaminated with or potentially contaminated with pathogens requiring a defined biosafety level. So these are the infections of special concern: tuberculosis, hepatitis B, HIV, enteric infections, and the routes of infections, inoculation, ingestion, and inhalation. The biosafety in microbiology labs preventing lab-acquired infections from bacteria, viruses, fungi, parasites, prions, and recombinant DNA is the source of virus specimens, human blood, unfixed tissue, human cell lines to the lab personnel and to the community.
So, there are two types of barriers, which are the primary and the secondary barriers. First, the primary. Physical barriers are personal protective equipments for lab workers. Secondary barriers are structural aspects of the laboratory that make working environment safer against infection. So, what are the universal safety precautions? Number 1. Consider all the specimen potentially infectious for HIV and other bloodborne infections. Number 2. All specimens should be placed in a leak-proof, impervious container for transport. Number 3. Use gloves while handling all samples, especially when there is contact with body fluids, non-intact skin or mucous membrane. Next, if there is likelihood of spattering, use face masks with glasses and gowns. Wrap-around gowns should be preferred. These should not be used outside the lab. Next, cover cuts or abrasions present over skin with waterproof bandage. Number 6. Decontaminate the laboratory work surfaces immediately in case of spillage of the blood or any other of fluids. Number 7. Follow no needle recapping strategy. Number 8. All sharps should be collected and disposed away properly. Next, never pipette by mouth. Use mechanical pipetting devices. Number 10. There should always be a system working efficiently for management of hospital-generated waste. Number 11. It is advisable for the laboratory personnel to be vaccinated against hepatitis B. And lastly, facilities should be available easily for post-exposure prophylaxis in case of exposure to HIV and HRV. And these are the don'ts and do's in standard microbiological practices. So next, we are going to the biosafety levels. Precautions to be taken by people researching or trying to identify organisms. Labs must adhere to these specific safety regulations. So, a biosafety level is the level of biocontainment precautions required to undertake while handling dangerous biological agents in an enclosed facility. And now, we are going to the levels of containment. First, biosafety level 1. Microorganisms that don't consistently cause disease in healthy adult. E. coli or polyomavirus. Basic laboratory, standard microbiological practices. So these are the standard practices required. First, frequent and hand washing, door that can be kept closed when working, limits on access to the lab space when working, no smoking, eating, drinking, storage of food, and laboratory. Care to minimize splashes and actions that create aerosols. The contamination of work surfaces after every use after any spills.
Biosafety Level 2, agents associated with human disease, generally required for any human-derived blood, bodily fluids, tissues in which infectious agents may be unknown. Agents include Physalis virus, Salmonella species, pathogenic, Toxoplasma, Clostridium, Botulinum, and Hepatitis V virus. Here are the additional information about our topic. Under BSL, we have 4 categories: their microorganisms and their remarks. Primary hazards: accidental needle sticks, exposure to eyes and nose. Agents do not cause lethal infections and are not transmissible via airborne route. Agents are pathogens for which immunization or antibiotic treatment is available. Extreme care should be taken with contaminated needles and sharp lab instruments. Next, standard practices include BSL 1+, policies to restrict access to lab, biohazard warning signs posted outside the lab, surveillance of laboratory personnel with appropriate immunization offered, biosafety manual with definitions of needed waste, decontaminations, or medical surveillance policies, supervisory staff who have experience of working with infectious agents and specific training of laboratory personnel in handling these agents. Next is the Biosafety Level 3, BSL 3, microorganisms that cause serious disease transmitted by inhalations, tuberculosis, yellow fever virus, hantavirus, Y. pistis, or plague, containment lab, double door entry, directional airflow, all work in biosafety cabinet. Standard practices include BSL 2+, strictly controlled access to the lab, specific training for lab personnel in handling potentially lethal agents, decontaminating all waste, changing contaminated protective lab clothing, decontaminating lab clothing before laundering, institutional policies regarding specimen collection and storage from workers to prevent exposure. Next is the primary barriers, biosafety cabinets, or other approved contaminant devices, personal protective equipment, lab coats, gloves, faces, protection as needed, protective clothing removed when personnel leave laboratory area, cabinets truly decontaminated daily and monitored for radiation for personal protections, secondary barriers, BSL1 barriers, plus autoclave for glassware. Next is the primary barriers, similar to BSL2 personal protective equipment, respiratory equipment, secondary barriers, corridors separated from direct access to lab access through self-closing double doors. All BSL2 barriers, air handling system to ensure negative airflow or air flows the lab. Next is the air pump into the lab, not recirculated in the building. Next one is Biosafety Level 4, BSL4 microorganisms that cause lethal disease with no known treatment or vaccine. Example of this one is Ebola virus and Marburg virus. Maximum containment lab, positive pressure ventilated suit or moon suits. Dangerous and exotic agents with high risk of life-threatening disease and are aerosol transmission. Related agents with unknown risk of transmission. Agents or all viruses include Marburg virus, Ebola virus, viruses that cause Congo Crimean, hemorrhagic fever, and Lassa fever.
Primary barriers, biosafety cabinets used at other biosafety levels, full body, air supplied, positive pressure personal suit. Secondary barriers, all physical barriers at BSL3, isolated zone or a separate building, dedicated supply and exhaust vacuum decontamination systems, and a recommended absence of windows or sealed and resistant to breakage. So let's now proceed to the new 8 categories of biomedical waste. Here are the categories with their type of waste and treatment and disposal. Category 1, type of waste, human anatomical waste, treatment and disposal, incineration or deep burial. Category 2, type of waste, animal waste, treatment and disposal, incineration or deep burial. Category 3, type of waste, microbiology and biotechnology waste, liquid waste, waste from laboratory, blood banks, hospitals, house, and etc., treatment and disposal, local autoclaving, microwaving, incineration, disinfection by chemicals. Category 4, type of waste, waste sharps like needles, syringes, scalpels, blades, glass, and etc., treatment and disposal, disinfection, chemical, autoclaving, microwaving, and mutilation or shredding. Category 5, type of waste, discarded medicines and cytotoxic drugs, treatment and disposal, incineration or destruction and disposal in landfills. Category 6, type of waste, solid waste items contaminated with blood, body fluids including cotton, dressings, and etc., treatment and disposal, incineration, autoclaving, and microwaving. Category 7, type of waste, solid waste like catheters and IV sets, and etc., treatment and disposal, disinfection by chemical treatment, autoclaving, microwaving, and mutilation and shredding. And Category 8, type of waste, chemical waste, treatment and disposal, chemical treatment and discharge into drains for liquid and secured landfills for solids. And these 8 categories are again sorted according to special color-coded receptacles for waste of those categories. The waste is collected in these color-coded containers and processed further accordingly. The following table depicts this categorization. So here are the color coding with their type of container to be used, waste category number, and treatment. First is yellow. Type of container to be used is non-chlorinated plastic bags. The waste category number are category 1, 2, 5, and 6. The treatment is incineration. Second is red. Type of container to be used is non-chlorinated plastic bags or puncture-proof container for sharps. The waste category numbers are category 3, 4, and 7. The treatment is disinfection, autoclave, microwave, mutilation, and shredding or landfilling. The next is blue. Type of container to be used is non-chlorinated plastic bags container. The waste category number is category 8 and the treatment is none. And the last one is black. Type of container to be used is non-chlorinated plastic bags. The waste category number is municipal waste and the treatment is none.
So here are the additional information and overall summary about our topic. And these are the references that are used in our presentation.
And that's all about our presentation of Activity 1 entitled Biosafety in the Laboratory. Thank you very much.